About metabolism, everybody's favorite subject, right? Those of you who have had physiology, remember Krebs cycle? Do you remember glycolysis? I heard the gas. Yeah, exactly. The ETC. What's that? We're going to do it again. We're going to do it again. So exciting. So if you loved it before, you're going to love it a second time. If it's your first time, yeah, you get to learn it with me. I will teach you the way that I kind of like pictured it in my mind. It might be a little bit different, but the process is the same. I'll just give you some simple ways to help keep track of everything. So some of it will be review from physiology. For my Tuesday, Thursday class, we are having our lab quiz tomorrow. I will be giving you back your lecture exams during the lab, so you'll get those back tomorrow as well. In case you're going, like, are we going to get those back? So you get them back tomorrow. Let's do them together. The exam, the lab exam will be the first hour of class, so from noon to 1, and then at 1 o'clock we'll do our lab. We'll be reading out our chapter, our experimentation. Okay, so we're going to be talking about how bacteria live, how cells are able to create energy, and how they're able to use metabolism to survive. So in these processes, one of the things about metabolism is that we have to constantly have an influx of nutrients for our biomolecules. So we learned about the 4 major biomolecules in our chemistry chapter. What were those 4 major biomolecules?
Carbs, lipids, nucleic acids, and fats.
Excellent. So carbs, lipids, nucleic acids, and proteins, right? That's okay. You said lipids and fats. Yes. I knew what you meant. So those are our 4 major biomolecules. We and bacteria, prokaryotes, have to have these to survive, and we have to have a stable input of these. So when we eat, or when we consume food, what we're doing is we're getting the building blocks to create new proteins, new carbs, new lipids, new nucleic acids. If we don't have this constant influx of fresh nutrients, then we're not able to do growth. So the growth requirements and the metabolic byproducts, so what we're going to create through our metabolism, help us organize our prokaryotes. We're going to learn that certain prokaryotes like to eat certain foods. We use that in the lab to help identify or key the different genus and species that we're dealing with. We can look at what they make as they eat to help us identify them as well. So they're all going to be essential. Now, one thing to remember is oxygen is essential for us and our metabolic process. For a lot of bacteria and prokaryotes, it is not essential for them. We're going to learn that some organisms are obligate anaerobes. We're obligate aerobes, which means we need oxygen. Obligate anaerobes will get killed when oxygen's nearby. We're going to learn why in this chapter. Also, we have some prokaryotes that are called facultative anaerobes. And what this means is they can go back and forth. So they can live with oxygen, they can live without it. The big difference though is they prefer oxygen. So they like to have oxygen, they can get more energy, but if it's not there, they can survive. And we see that with a lot of, like, for example, our gut flora. E. coli, we grew E. coli on our spread plates. So it can grow with oxygen. But E. coli also lives in our guts, and our guts are anaerobic. So it can live either way. It's fine. So metabolism is basically the sum of all of the chemical reactions occurring in an organism. So what that means is we're bringing food in, we're breaking it down, and we're processing it. So energy is often going to be transferred from one molecule to another. And what I mean by that is we're going to be doing what's called oxidation and reduction reactions. So oxidation and reduction is where we're transferring or we're moving electrons from one molecule to another. as we break things down or build things up. So one of the ways to remember this is a couple of different mnemonics when it comes to oxidation and reduction. What's one mnemonic that you all have heard?
Oil rig.
Leo-ger.
Okay, so I've heard oil rig and then Leo-ger. Is that what you guys have heard too? Okay, so oil rig stands for oxidation is losing. Yeah, so what that means is it's losing an electron, okay? And reduction is gaining, yeah. Okay, so oil rig oxidation is losing. So if I say something's been oxidized, that means it has lost electrons. If I say something has been reduced, that means it's gaining electrons. Now, if you're like me, you might be looking at this going, whoa, whoa, whoa. I'm reducing while I'm gaining? What's going on here? Like, that doesn't make sense. Those seem like they're opposites. What they're referring to is the charge. So if I gain additional electrons, what happens to my charge? It goes down. It becomes more negative. So my charge is what's really being reduced. All right, I'm getting more negative. I'm getting more electrons than I have positive protons. So that's why I'm getting reduced. We have Leo-ger, so that stands for losing electrons is oxidation, and gaining electrons is reduction. So that's another way you kind of picture that. Whichever one you prefer is fine with me. I don't care which one you use, just as long as you're keeping track of it in your head, because when we start talking about things getting reduced and oxidized, it can get a little confusing. Now, the one thing to think about with oxidation and reduction is they always go hand in hand. If somebody's losing an electron, somebody has to be gaining them. It's kind of like a teeter-totter. If somebody's going up, somebody's going down. All right, so I can't do one without the other. So we refer to those a lot of times as redox reactions. And what that means is it's an oxidation and a reduction. They're happening simultaneously. Somebody's losing, somebody's gaining. That's a redox reaction. All right, so that's used to say that this is occurring. So with that then, electrons transferring is going to be part of our metabolism. Yes?
Okay, so I know from like, physiology.
Yes.
They were talking about like oxidation has to do with hydrogen.
Uh-huh.
Was it the same here?
Same thing here, yep. So the question was, I know in physiology when we talked about oxidation and reduction, it was usually to deal with hydrogens. The same is gonna be true here. We're talking about electron flow and electrons being removed. Electrons, in the case of metabolism, that are being lost or gained, it's usually hydrogens that are losing them and somebody else is gaining them. And the reason hydrogens can lose their electrons somewhat easily is, let's think about our friend hydrogen. He's got a atomic number of 1. So how many protons does he have?
1.
1. And how many electrons?
1.
1. And is he happy with that, or is he unhappy with that?
Unhappy.
Unhappy. How many more does he want in the show?
1 more.
1 more, or he can get rid of the electron. So a lot of times what they'll do is they'll just kind of like, boop, give that electron away, and then they'll become a positive ion, and then somebody else will pick it up and become more negative. So if that happens, what happened to the hydrogen? Was it oxidized or reduced? It was oxidized, right? Oxidation is losing because it gave up its electron. And whoever took it was reduced. So yes, so you are absolutely correct. We're gonna be getting there. When we start talking about things, we'll be talking about electrons flowing and hydrogens. So we can have reduced and oxidized forms of different elements that are out there. For example, reduced forms of carbons. They carry a lot of potential energy. So if they're reduced, remember that means they have a few extra electrons, which means they have a lot of energy behind them. So things that can be reduced are things like hydrocarbons. What do we use hydrocarbons for? Fossil fuels and energy, a lot of those are hydrocarbon based. So like just living our normal lives, driving our cars, they can use hydrocarbons. Methane is something that we can also use as energy. Fats and carbohydrates, do they have a lot of energy?
Yes.
Yeah, I mean, they're really good energy. Alcohols, does alcohol have a lot of energy in it? It actually does, yeah. Have you ever heard of somebody having a beer belly? Yeah, we get those beer bellies because when we're drinking a lot of alcohol, we're bringing in a lot of carbs and we're bringing in a lot of energy. And then if we're not using it, that extra energy starts to get carried around. So that becomes our square belly. I just called it the keg. Right? You start carrying the keg with you. It's like you start getting the beer belly. Now, we also can have oxidized forms. So remember, oxidized means that we've lost our electrons of carbon organisms. So this can be things like our ketones. What do we think of when we hear ketones? Yeah, ketoacidosis, diabetes, yeah, we think of, ketones aren't that great, we don't want a whole bunch in our body, they can lead to acidosis. But they also are a low energy level, so it's something that's not really helpful for us. Carbon dioxide is also something that tends to have a lower energy state. They just don't have a lot of potential for chemical energy, so a lot of times they're wastes. They're products that we're not relying on as an energy source. Now, when we start talking about metabolism, we're going to find out that it is very heavily regulated by our enzymes. So enzymes are going to be huge in metabolism. They're going to be what's going to help drive each step in the process. So every step along the way has a set enzyme associated with it. And we'll learn the ones that are important. There's certain ones that I'll want you to know for your exams because they're going to be important and come up later when you go on to your nursing profession or whatever profession you go into. The thing to think about metabolism is it's basically just a series of steps. And each step that's done will lead to the next step. It's just like when you're walking downstairs. You take the first step and you work your way down. Hopefully, don't fall. Anybody in here a frequent faller? I always panic, like my office is upstairs, my classroom's downstairs. When I walk down those steps and it's in front of everybody, I'm like, don't fall, don't fall, don't fall. It's always in the back of my head, don't fall, don't fall. Okay, so if I tell you a molecule is reduced, what does that mean has happened to it? It gained electrons, good. And I don't have the answers for these right in here, I apologize, so it'll be gains electrons. There's no highlighted answer, it's gonna come up. What is meant by oxidation? Or losing electrons. I have it here on the board. If I want to be a cell and I want to grow, what are some minimum requirements? What do I have to bring in? Nutrients, energy. Yeah, I have to bring in those amino acids. I have to bring in those carbohydrates, those biomolecules. And I have to be able to convert them into a usable form. And then just in general, if you were trying to explain what metabolism was to somebody, what would you say is a general description? It's the sum of chemical processes in the body or in the cell. That's great. It's the sum of all reactions in a cell. So a cell is always very efficient. They're not going to waste their time. We do metabolism because we get something from it. We get energy. So if we're not getting energy, we're not going to waste our time with that.
The third one was energy in, is that what you said?
Yes, so biomolecules in.
Okay.
Yeah, so I have to have constant energy and constant biomolecules. And I do apologize, I will put the answers in the future. I just didn't get a chance to do it on this slide. I was actually sitting in my budget meeting going, I didn't update that slide. So I was like, I felt guilty, but I didn't know. Are we good with this before we move on? Okay, I'm sorry, they will be there in the future. So before we go into metabolism, let's get a little bit more familiar with some of the key players. So again, the key players that kind of run and regulate this whole process are enzymes. So we've talked about enzymes before. What type of biomolecule are enzymes? Proteins, excellent. And the whole purpose of an enzyme is it's gonna increase or facilitate a reaction. It's gonna allow things to happen. Now, when you picture an enzyme, I want you to kind of picture a lock and key analogy. It's just like our front door key. My front door key can go into my front door lock, it can turn it, and the change that I see is my door will open. Now, do I break that key and that lock each time I use it? No, it's reusable. The same is true with enzymes. So I'm gonna have a key go into this enzyme. The enzyme is like the lock. If the key fits into the enzyme, a reaction will happen. So if this key fits into the lock and I turn it, my door will open. Now, I think we all have car keys. What happens if I try to use my car key on my front door lock?
Doesn't work.
It doesn't work. I've tried this when I'm really tired. You pull out the wrong key and you're like, why isn't it work? Oh, never mind me. Yeah, if I try to use my front door key on my car, it's not gonna work. It has to fit into the lock. So with enzymes, they're very specific. I have to have the right key to unlock me. Okay, so that's a very specific thing. The other thing about enzymes is just like the lock on our door, again, they're not destroyed. They can change, they can cause a reaction, but they stay there. So we're not losing them when we use them. Whoever decided to name enzymes, I wish I could go back in time and give them the hugest hug. Because most enzymes end in ASE. That means I'm an enzyme. So for example, if I'm eating lactose, which is a sugar, what do you think the enzyme would be that would break it down?
Lactase.
Lactase. Yeah. Oh, so easy. If I have glucose, what do you think the sugar would be that would break it down? Oh, you would think glucose, but no, it's amylase. Oh, why? Why did they were so close. It's because used to glucose was called an amylose. That was the Latin name for it. People decided amylose was too hard to remember, so they switched it to glucose to make it more like a glucogen and glucosides and sugars, but they forgot to change the enzyme. So, yeah, we'll get there. But for the most part, ASE is how you can think of your enzymes. Now, of course, there are a few exceptions, but that's the general rule. So remember, there's a few enzymes out there that don't end in ASE just to make life difficult, but 98% of the time they're ASE. Now, with an enzyme then, again, if something fits with it, we'll get a reaction. So what's gonna fit with the enzyme? Well, we're gonna refer to that as a substrate. So the substrate is the key that's gonna unlock that enzyme. Okay, so the enzyme's the lock, the substrate's like the key, and then the product is what we end up with from their interaction. So it's just like, you know, putting the key into the lock, turning it, we get the door open, that would be my product, is the open door. So with this then, enzymes work really, really fast. The turnover number is about 1 to 10,000 molecules per second. So imagine opening 10,000 doors in 1 second. That's basically what these enzymes do. So they are super efficient. They will get a process done very quickly. It's pretty amazing how quickly they work because an enzyme is always present. It's just waiting for something to bind with it and to make it active. And so you think about all the things that are constantly bombarding enzymes, but we hit them hard enough that we actually get reactions most of the time. I mean, 10,000 in a second is pretty amazing. And so then what can happen is my enzyme and the substrate, the lock and key, come together, and then what gets released, the product, will be a little bit different. And that product then can go find another enzyme, and then that enzyme can work with it. So for example, let's say I'm bringing in some food. My first step with this food is maybe to have this protein come in and break it to an amino acid. Okay? So if I'm breaking something down, what kind of reaction is that? Is that decomposition, hydrolysis? Hydrolysis, yeah. So hydrolysis is going to happen. So my enzyme's going to grab it. It's going to help break apart this protein into an amino acid. Now, I grab it, I break it, and now my product that's released is 2 separate products. These products can now go interact with another enzyme, and so on until I get the final result that I want. Does that make sense? So each product that gets released usually has another enzyme nearby that's ready to grab it and use it. So, as I said, enzymes, they usually are going to make sense as far as their names go. So for example, if I say that we are looking at hydrolases, that's a group of enzymes, their job is to add hydrogen and hydroxide in the form of hydrolysis. So they're helping to move hydrogens around. Hydrolases, makes sense. Lysases, if you hear something as a lysase, that means it's splitting, it's lysine. When you hear isomerase, that means it's rearranging to balance, so structural rearranging. Ligases, it's like a ligand holding something together. So a ligase is going to join molecules together. Oxidoreductase, oxidations and reductions. That's a long word. And then transferases, they're going to be moving or transferring functional groups. So again, these are kind of just the general categories, but their names hopefully make sense, right? So hydrolases, lysases, ligases, and transferases are the ones that we'll be talking about the most in this class. And then also probably some oxidase bases. But those are just their categories, that's just what they do. And then we'll learn specific ones along the way. So the whole idea behind enzymes is that they're always helping us. They're always our assistants. So there's this idea that we have something called activation energy. And I apologize, my beautiful picture is kind of over this, huh? Let's just squeeze this guy down. Let's make this slide a little prettier. I think your slide in your notes is probably pretty, right? Hopefully it's pretty. All right. Oh, that looks so much better. Okay, so activation energy is going to be the amount of energy that I need to put in to make a chemical reaction happen. So in other words, what energy do I need to fulfill to get something rolling? Picture like a ball rolling down a hill. I have to push it to get it started. The reaction rate is how frequently something would occur if I had that energy in place. The enzymes are going to take that reaction rate and they're going to speed it up. So let me explain here. I am having some balls on a billiard table, okay? And I want this ball to go into a corner pocket. What can I use to make those balls move in billiards? I can use a cue stick, and which ball am I gonna hit? The cue ball, the white ball, okay? I hit the white ball and it's gonna hit other balls out of the way, all right? The white ball is like my enzyme. It's going to hit other balls and make other reactions happen. There's a little bit of energy that goes into getting that cue ball to get started. That's my stick, right? I have to hit it. But once it goes, it's gonna make other processes happen. So with my enzyme, what it's gonna do is it's going to take that activation energy and it's gonna reduce it. It's gonna help bring things down. So the chances of things bumping into themselves in the body or in a cell, well, by chance, things could randomly bump into each other. An enzyme helps guide that bumping. So we kind of guarantee that it's gonna happen. So the activation goes down. We get a little less. The reaction rates, the chances of things encountering, go up. Okay, so activation drops, reaction rates go up. Another way you can think of this is think about a matchmaker. Oh, eHarmony, right? Matchmakers go through and they weed through all of the ones that you don't want to be with, and they try to find the ones that you do want to be with, okay? That's kind of like what the enzyme's doing. So when you're on eHarmony, your activation, your energy of going out and meeting somebody drops. Because they're doing that for you, they're scanning for you. Do they still use eHarmony? I don't even know anymore. What do y'all use now? What's the spiking one? Tinder, there you go. Yeah, it helps too, right? It kind of drops the, I don't know. Yeah, it kind of drops the activation. You're just sitting there swiping. I mean, that's low activation. Okay, we drop the activation. How am I going to meet it? That's my reaction rate. Okay, the chances of coming across somebody is what's being put on my thing. Does that kind of help explain that a little bit better? Maybe a little too much. It's a little TMI, right? Okay. Okay. So many proteins and enzymes that are out there, they're able to work, but they're incomplete on their own. We kind of actually do this on purpose in the body because what we don't want to do is we don't want to have an enzyme that's just kind of sitting there if I don't need it. So for example, bacteria and prokaryotes do this really well. They turn enzymes off, which is pretty cool. So for example, I said lactase is going to break down what? Lactose. Okay. But if I'm a bacteria and I'm sitting in a solution and there's no lactose sugar around, do I want to put in energy to make that enzyme? No. So I'll just turn my enzyme off. I won't even worry about making it until lactose is around, and then I'll turn it back on. The same is true with a lot of the enzymes in our bodies. We don't necessarily shut them off, but what we do is we have enzymes that are incomplete until we get all the pieces present. And then once all the pieces are present, then we can build the enzyme and make it work. So, for example, an apoenzyme is going to be a segment, it's a non-active protein portion that needs to have something else present to activate it. Now, things that can activate it can be things like cofactors. Cofactors are things that we get from our diet. So, for example, if we don't get iodine, what are one of the side effects that can happen if we don't have iodine?
Goiter.
Yeah, we get a goiter, we get a swelling on the neck. That's from the thyroid starting to swell up. So the reason that we have a goiter is because if we don't have iodine, we're not having enzymes function properly. So an apoenzyme is an enzyme, but it's not activated yet. I get a cofactor from my diet, and it's gonna fit with that enzyme, and it's gonna activate it. Okay, and now when I activate that enzyme, now it can work. When the enzyme gets activated and it's ready to go, we refer to it as a holoenzyme. An easy way to think of this is a whole enzyme is a holo enzyme. Isn't that a little easier? Holo. Yeah. So the idea behind this is when I have my two factors that I need to make the enzyme work, it's holo, it's whole, and now the enzyme can function. So I'll show you a picture of this. Here's an apoenzyme. So this is an enzyme that is incomplete, okay? So it's not gonna work, it's not gonna function, it's ready, but it needs some help. Then I can have a cofactor come in and it will bind with my enzyme. And now my enzyme is ready to go. So I refer to that as a holoenzyme. Now this enzyme will function. And now it's ready to receive those keys and have those locks unlocked, all right? So apoenzyme means I'm not ready yet. A cofactor has to come in and turn me on, and then I can become a holoenzyme. And cofactors can be a lot of different things, but they're usually additives in our diets. So for example, if you're having a low metabolism, one of the things that they'll recommend is that you start ingesting what vitamin? Which one? Vitamin B. Vitamin B is a big one. So vitamin B helps boost metabolism. The reason vitamin B helps, it's one of our cofactors that helps with our enzymes. Another one is vitamin D. Vitamin D is a cofactor for our immune system enzymes. So there are certain things that we can take that help boost our enzymes. All right, so if you have slow metabolism, try taking a multivitamin B. There's different vitamin Bs that are out there. So with this then, our enzymes are going to be really important in metabolism. Now, enzymes can also help with redox reactions. So remember we said that that's where we have one element losing electrons and somebody gaining them. The enzymes can help with this because a lot of times they act like little shuttles. And they're going to help shuttle electrons around. So the way we'll talk about these when we start going through metabolism is we'll refer to these special cofactors or enzymes as Uber drivers. They basically are going to pick electrons up and they're going to drive them to a different location in the cell and drop them off. So a couple of main hitters that we're going to talk about in metabolism are going to be our cofactors that are NAD and FAD. NAD and FAD, we'll talk about them in a few slides. Their job is basically to pick up electrons and to move them later in the cell. They're going to be really important when it comes to the electron transport chain. So that brings back memories, the electron transport chain is going to be centered around these NADs and FADs. Now, NAD can pick up a pair of electrons, and the electron donor, who is hydrogen, will also come along for a ride. And so when NAD is picking up passengers, it becomes NADH, okay? So what that means then is NAD is the oxidized form, NADH is the reduced form, all right? So the way you can picture this is picturing, again, Uber drivers. If I'm NAD or oxidized, that means that I'm ready to pick up passengers. If I'm NADH or reduced, I have no room. My Uber is full. And I have to drop off those electrons and those hydrogens first, and then I can pick up new passengers. All right, so NAD and NADH are one of our major FAD will turn into FADH. It'll actually turn into FADH2. Don't worry about it. I'm not having you guys trying to keep track of all the electrons. Basically, again, FAD means I'm open for passengers. FADH means I'm closed for passengers. I'm not taking any more. All right? And we'll be talking about a lot of these when we go through our metabolism. So their full names, if you wanted to know, I have them up on the screen. I will never ask you these names. If I ask you a quiz or a test question, I will always refer to them as NAD or NADH or FAD or FADH2. So I won't ask you these full names. Again, this is something that you'll learn in biochemistry, but to me, it's the acronyms that are important and knowing what part they're playing as opposed to knowing their full name. Just like DNA. Does anybody ever come to you and say, do you happen to have any deoxyribose nucleic acid? And you're like, no, people use DNA, right? So nobody ever says these long names. I mean, you might have a professor who's like, who cares? NAD, NADH, FAD, we're good, all right? We'll talk about their players, roles, when we go through metabolism. Now, the one thing I do want to remind us though is when we're picturing these things, sometimes in lecture, it's hard to think of enzymes as being 3D structures. So I have this picture here just to remind us. When we're going through the flow and the processes that we're going to cover today, it looks like this nice linear, very straightforward fit. But remember, enzymes, they're in a cell, so they're actually 3D. So when you picture an enzyme and picturing this lock and key, it's not a flat reaction. It's this whole mechanism where everything has to fit just right. Remember in chapter 2 when we talked about protein folding, we talked about how we had our folding that could be quaternary or tertiary or secondary. And I told you the folding dictates whether or not we're an oven mitt or a baseball glove. Remember those whole stories. The same is true with these enzymes. How they're folded is going to dictate how they're going to react. Okay, so folding is going to be really important here when we talk about our enzymes. Now, enzymes are great, but they are a little bit sensitive in certain places in the cells as far as their environment goes. So, for example, enzymes can be very sensitive to temperature. Now, what's interesting about enzymes is if you increase the temperature a little bit, they actually work a little faster. But if I go too high, I can break them. And if I break them, I can start to do something that's called denaturing. And denaturing means the enzyme's starting to fall apart. My 3D structure is starting to melt, okay? And if I push it too far, I can irreversibly denature it. It can't recover. So for example, if I have a fever and my body temperature gets too high, when I first start to get a fever and I'm in the 99 to 100 range, my enzymes are actually working more efficiently. They're working harder with a low-grade fever because the temperature's gone up a little, and that actually gives a boost to my immune system. So people think of fevers as being bad. When it's a low-grade fever, it's actually not bad. That's your body protecting you. What temperature do you start to get worried about fevers? 104. 104, yeah. And the reason we get worried at 104 is because 105 is getting to a critical range. If I get to 105 or, God forbid, 106, I'm getting into the denaturing range. So I'm getting to the point where enzymes are going to start melting. Do I need enzymes to function? Oh yeah, I need enzymes to function. So I have to be careful with my temperatures. If I get too hot, I could have an irreversible denaturing. This is what we see with heat strokes, or when we have damage where our bodies have gotten burned. The flip side of this, we can also slow our enzymes down with temperature. So if I put somebody in really cold environments, their enzymes actually slow, and they stop working as efficiently. This is something that's actually used on accident victims. So one of my students was telling me this story. She was involved in this really bad head-on collision accident, and she was trapped in her car. She said, she was really stuck in there, and they had to use the jaws of life to get her out. And she said she was sitting in her car, trying not to panic, feeling a lot of pain. She had hurt her back and neck. And she said that finally they get her out of the car and she's just like, oh my gosh, thank heavens, the worst part's done. And she's like, they put me in an ambulance and they put an ice cold blanket over me. And she's like, I'm like, put me back in the car. This is worse. You know, now I'm freezing cold. Why did they put an ice cold blanket over her? They were slowing the enzymes down. And why were they slowing enzymes? To slow down shock, to slow down blood flow, to slow down clotting. They're slowing things down to try to have time to get her to a hospital where they can then heat her back up and get things back under control in a controlled setting. So this can happen. So you might have heard stories of people that were like underwater for 5 minutes and then they were recovered and they were fine, no brain damage. And you might be going, how did that happen? I guarantee you it was ice cold water. Everything kind of slowed down. Their body went into a little freeze for a little bit. So our enzymes can do that. They can kind of adjust. Another one that can affect enzymes is pH. So if I eat something that's too acidic or something that's too basic, that can affect my enzymes. So again, enzymes can be affected by pH. Let's think about our stomach. What kind of pH do you think that our enzymes in our stomach like?
Low.
Acidic. Yeah, they like low pH. But then we go into the colon, and the colon has a higher pH. It's more basic to neutralize those acids. So those enzymes each have their own preference. Some like to live in acid, some like to live in a basic environment. Another thing that'll affect them is substrate concentration. So substrate's what's reacting to the enzyme. If I have a lot of chemicals coming in, my enzymes are working fast. If I only have a few chemicals coming in, the enzymes will slow down. And then inhibitors. Inhibitors help us control our enzymes. So we're going to learn about how things can be controlled in our enzyme world so that we can regulate what we're making and what we're doing. So here's a picture showing an enzyme that is getting denatured, and then a picture showing a food with irreversibly denatured enzymes. So when we cook eggs, we're adding a lot of heat. That heat on that egg is irreversibly denaturing the enzymes. And that's why we see the egg change in how it looks and its consistency. It's because we have broken all of the proteins down. Now, the way we can regulate our enzymes is through a couple of processes. And I know this is a lot of words, but we'll look at a picture that'll help make it a little bit clearer. There's allosteric inhibition, and there's the non-competitive inhibition that is the same thing. Basically, the idea is that I have an enzyme. You guys have to look at my horrible drawing here. I have an enzyme, okay? Looks kind of like a tulip, right? And this is the site where the substrate is going to want to bind, okay? And it wants the enzyme to have this shape. But I also have this little area at the other side of the enzyme that's referred to the allosteric site. And what this is this is an area that I essentially can turn, yes?
Use a different color. It's hard to see
Is,
that.
I don't know, let me see. My pins up here are very limited today, but we'll see, yes. Can this one, is that, oh yeah, okay, this one's right, okay, so. And I'll leave them up too so you can take pictures. Thank you. Okay, so this is going to be our allosteric site at the bottom of the enzyme. And up top is where the substrate's going to be. Is that a little bit better? Okay, good. I'll put substrate. Thank you for telling me. Okay, so the way this works is I can have regulators that can come in and they can bind at this allosteric site. And when they bind, they change the shape of the enzyme. Maybe not that dramatically. There, they change the shape of the enzyme. Do you see how my enzyme has kind of closed up a little bit? This is kind of like taking a key and sticking it in the lock and leaving it there. If I try to come in with the next key, can I get it to fit? There's something blocking. I can't get it to work. My lock has changed. So the idea behind allosteric site regulation is I have something that will bind to the enzyme that will turn it off. Now this is referred to as non-competitive because it's not binding where the substrate binds. It's binding in a different location. We basically have a separate spot for this regulator to bind. Now, the beauty of this is I can turn these enzymes on and off then by regulating the allosteric site. So for example, let's say I want to make some product. Okay, so I have the substrate coming in, it is interacting with the enzyme, and then product gets released. Okay? But let's say I've made enough product. I'm at the point where I don't need any more product. Do I want to keep wasting my time making more and more and more? No, this is like, if you go shopping, do you want to buy 5 gallons of milk? Where are you going to put those 5 gallons, right? We only have room for maybe 1. So I don't want to waste my energy or my time making too much stuff that then I have to store. So I'll use my allosteric site regulation so I don't make any more product for a while. And then when I use the milk or I use that product, when I get low, I can turn it back on. Does that make sense? So I can do this regulation. So again, it's non-competitive because it's not binding where it is where a substrate will bind. Now this is also referred to in physiology as a feedback loop, and in particular as a negative feedback loop. A negative feedback loop is when we're going down a path and then we hit what we need, so we stop and we go back. Negative not meaning bad, just meaning that we stop and we reassess, okay? That's different than a positive feedback loop where we keep going down the path and we never stop, okay? So negative feedback loop is where we get processing. So here is a much better picture than what I could ever possibly draw, showing that substrate coming in and binding at the active site of the enzyme. That's the lock and key area. And when I want to turn it off, I will have a regulator bind at this allosteric site, and that regulator will change the shape of the enzyme and turn it off. And then I can drop that regulator off when I want to turn the enzyme back. So that's allosteric regulation or inhibition. Now I also can have competitive inhibition. And in this case, instead of using a separate site, I'm just going to have my regulator bind in the same site as the substrate. This is competitive. They're competing to bind the substrate. The difference though is that when my inhibitor arrives, it won't move. So again, the analogy is kind of like picturing something in the lock. I can't get my key in there anymore. So I essentially just shut it down. Now, with this then, the enzyme will stay turned off until that regulator deattaches and lets go, and then the enzyme can start working again. So in our cells, a lot of times our regulators are the products that we make. Let's say I'm making a product. As soon as I get this product build up, it goes and shuts off the enzyme. I start using that product, and eventually I need to use that last product that's bound to the enzyme to shut it down. And when I pull it off, the enzyme turns back on. Isn't that beautiful? It's like, it's holding a spot, and then it's kind of like if you go shopping at Costco and they sell you the display model. The display model is like the regulator. Does that make sense? That's the last one. And so now they know the display model's gone, we gotta get more. Yeah? Okay. So with this then, the substrate will turn off, the enzyme will turn off if something's blocking that substrate. Now we use this a lot to help us. For example, remember our friend penicillin. We talked about which one was it able to work on, gram-positives or gram-negatives? Gram-positives, good. It blocks the enzyme that gram-positive bacteria use to build those thick peptidoglycan layers. So I block their enzyme with penicillin. So penicillin is coming in, let's say this is their enzyme. Penicillin is coming in and binding in the same location that their normal substrate would bind. Is that competitive or allosteric inhibition? Competitive inhibition, good. So the penicillin is competing for the same site. And the penicillin binds that enzyme and it turns it off. And now the bacteria cannot make cell walls. And if they're gram-positives, they fall apart, okay? The same is true with ibuprofen. When we take ibuprofen is blocking the pain receptors in our brain. Same with Tylenol. Ibuprofen also will block receptors for inflammation, so it helps stop inflammation. So So with pain, when something's not feeling right in the body, our body keeps sending signals to the brain. It's like having a toddler going, hey, 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 hey. And it's not helping, right? It's like your brain's like, I know, we're working on it, give me some time. If I take an ibuprofen or a Tylenol, it stops that, hey, hey. So then I can get some peace, and then I can start fixing things. The other ones that work are Lipitor. What's Lipitor used for?
Cholesterol?
Cholesterol. Yeah, Lipitor is used for cholesterol. So Lipitor is another drug that is going to be able to bind to our enzymes, and it stops cholesterol production. So we get a decrease in cholesterol. Now, Lipitor has some goods and bads. The bad thing about Lipitor, have you ever heard of people who had leg cramps when they took Lipitor? It's a side effect in a small percentage of the population. The problem they had was their enzymes for muscle relaxation were shaped similar to their enzymes for cholesterol. It's a biochemical thing. It's not all of us have it, it's just a few of us have it in our DNA. So for some patients, when they took Lipitor, after they were on it for like a week or two, they'd start having really bad leg pain to the point that they couldn't even walk from like this classroom to the lab without feeling cramping and exhausted. And the reason for that was because of their biochemistry. Lipitor was binding to their muscle cells and making their muscle cells constantly contract. Now, it was a low-grade contraction. It's not like you're like this and you can't move your leg. No, it's very low-grade, but the muscles couldn't relax. And you would have these leg pains because they were just tired. So you'd have to go to a different drug that didn't interact. It's one of those things that with some drugs, this is why some drugs work for some people, they don't work for others. It has to do with how our enzymes are playing with those drugs. Cancer drugs like chemotherapy, they work by stopping enzymes that help with mitosis, which is cell division, by regulating it. So as I mentioned, Lipitor is used to reduce cholesterol, but again, we have to be careful to make sure that Lipitor fits in just the cholesterol enzyme and not in our muscle enzyme. And again, that is just completely genetics. So the reason I bring this up, a lot of us have parents that are starting to age. If you have a parent who's on a drug and they're like, hey, I'm having this kind of weird reaction, don't ignore them. Like maybe look into it or kind of help them check the side effects. Because sometimes it can lead to serious side issues that might be something that's unique to their chemistry. So you never know what you'll come across. My mom, when she was alive, she was taking insulin for diabetes. And she had this, I can't remember which one it was, she had one insulin injection that made her sneeze nonstop for like 20 minutes. After every injection, she'd start sneezing. And she was over visiting with me and she gave herself an injection and started sneezing. And I'm like, what is this? And she's like, oh, I don't know, it just happens every time I do a shot. And I'm like, would you stop? Like, we're gonna get on a different drug. Like, there's a bunch of different insulin types out there. You don't have to deal with sneezing for 20 minutes. Like, that's not normal. Like, let's move on. Okay, enzymes. They increase the speed of a chemical reaction. So let's go through these each. Do they lower the energy of activation?
Yes.
Yes. Remember, that's the energy that I'm having to expend. That's like being on eHarmony and they're sending me matches. Okay, so my activation lowers. Do they increase or decrease the frequency of collisions?
Increase.
They increase. Good. So just to recap, I lower activation, so you're going to circle A. They lower energy activation. They also increase, so you're going to circle C, increase the frequency of collisions. So they make it less effort and more likely to engage. Okay, just like a dating site, less effort, more likely to engage. Okay, so circle A, circle C. I apologize for not having them highlighted. I feel like the worst professor today. So circle A, circle C, and we'll move on. What happens to an enzyme when it is above or below its optimal temperature? C, denatured. I like vanquish. Don't you think we should use that? I vanquish you. I love that. But no, you should circle C. It is denatured. And what is allosteric inhibition? Is C. Good. I'm binding in a different location for the substrate. As I drew so beautifully on this board. I mean, well, that's a whole masterpiece right there, right? So So on this page, for the first one, you're gonna circle C. For the next one, you're gonna circle C. Okay, let's start talking a little bit more about energy production and ATP, because that's the whole point of our metabolism, is getting energy. So the big picture in the bottom line, we're bringing food in, and when we get these food molecules, the first thing we're gonna start doing is oxidizing them. What happens when I oxidize? Lose electrons, good. So I'm going to oxidize my food, and I'm going to pull electrons off. These electrons are then going to be shuttled by Uber drivers to different parts of the cell, and I'm going to use those electrons to make some energy. Now in a cell, what do we call energy? What's its acronym? ATP. So I'm going to make it some ATP with this process. All right, that's going to be my goal in doing metabolism. Now ATP is going to be where I add phosphates onto adenosine diphosphate to create ATP or energy. So we mentioned this before, back in chapter 2, that ATPs are energy token. The way ATP works is I could shoot one of those phosphates off, and that phosphate's like a cue ball in my billiards. It goes and does other energy. When that happens, I go to ADP, adenosine diphosphate, and then I wait to get a phosphate put back on to go back to ATP. We mentioned that we can go to AMP. How many phosphates does that have? One. But do we like to do that?
No.
No, it's way too much energy to try to stick 2 more phosphates on. Way more energy efficient to go ADP to ATP. The only time we really ever go to AMP is when we're in crisis mode, like running from a bear, running for our live fireflies, all right? That's really the only time we're gonna risk going that low, because we're gonna need a long recovery rate to build that, okay?
Can you go back one slide?
Yes, please, this one, I'm so sorry.
Thank you.
I get all excited about metabolism. Plus, I had a huge espresso coffee this morning. I have to do one. Which I think y'all noticed in the lab, I was like throwing their lab exams out to people on my Monday Wednesday lab. So bad, it was like passing around, I'm like, shit. It was like, like aggressive passing. It was like, oh, calm down. Okay, are we good on this one now? And thank you for asking, because like I said, if you're thinking other people are still. Are we good on this one? Okay. So in this process then, I'm going to get some ATP that's going to be generated, but I'm going to have to use a little work to stick those phosphates back onto that ATP. Remember, phosphates naturally have a little negative charge. What do we know about like charges? Do they like to be together? No. So it takes a little effort to stick phosphates together because they're not super thrilled about that arrangement. I mentioned before, it's like having 3 kids in the back seat. They would really prefer to only have 2. So it takes a little effort to shove that 3rd one into the square box. That's basically what we're doing. So with the whole process of ATP, we're going to find out that the biggest bang for our buck is going to come from the electron transport chain. Now, we refer to this part of ATP creation as oxidative phosphorylation. Holy Toledo, that's a mouthful. What that's saying is it's saying that something was oxidized. What does that mean? Lost electrons to give me the energy to add a phosphate to ADP. Does that make it a little bit more acceptable? I'm kind of breaking that down. Yeah, so for example, when I eat, as I said on the previous slide, my food molecules are going to be oxidized. So I'm oxidizing something. And then I'll be using their electrons to make energy. So that's what oxidative phosphorylation is. It's me eating and converting to energy. And that's going to be the big bang for our buck in the electron transport. So ATP, or the process that's going to make ATP, it's going to be essential for our survival. Now, with ATP, we're going to have processes that are going to release energy, and we're going to have processes that are going to use energy. When we look at energy production, one of the things we're gonna talk about is this concept of respiration, respiring in the cell. Respiration is the chemical formula that's shown here. And again, it looks scary, but let's break it down. C6H12O6. We've seen that a lot. What is that? Glucose. Yeah, so above C6H12O6, you can write glucose. So I'm going to take some glucose. What else do I need to survive? Oxygen. I'm going to take some oxygen. So I take some glucose and I take some oxygen. My waste products that I'm going to make, the waste that I get released, what do I breathe out? Carbon dioxide. So there's my CO2. I'm going to make some water. That's going to be released. So that's going to be some waste. And I'm going to make some energy. I'm going to make some ATP. Okay. The way I'm going to make ATP is I'm going to be adding phosphates to ADP. Okay. And I have to do a little effort. I have to put a little energy in, but I get a lot of energy out. So that's my ATP that I'm going to be getting out. So that's going to be the basis of respiration. Y'all want to know something really fun? How does photosynthesis work? It's the exact opposite. This is just a fun note. I won't ask you this on test. But photosynthesis is CO2 and water to create glucose and oxygen. What? It's almost like we grew up together. Yeah, life forms have adapted with each other, which is pretty cool. So we do the opposite.
So what is the first equation and versus
So the
second
first
one?
equation is what's going to happen to us when we eat. That's called respiration.
Oh, okay.
And so that's glucose and oxygen is going to be converted to CO2, which is a waste product, some water, and most importantly, energy, which is going to be ATP. And then that's showing the way we get ATP is to phosphorylate ADP. Okay, so we're going to go through these processes step by step to make sure that we're comfortable with them. We're going to start talking about glycolysis, how sugar is made and flows in the body, or how it's used and flows in the body, and in a bacterial cell. And we're going to find out that prokaryotes and eukaryotes do these processes very similarly. So it's pretty nice. That's why so much of this is review and physiology. You learned about human metabolism. Guess what? Prokaryotes, they mostly do human metabolism. We're very similar. So we can think of metabolism as happening in three main events, or three main stages. And this is true whether I'm a prokaryote or a eukaryote. My first stage or first event is glycolysis. Okay, let's break this down. Lysis means what? Breaking. Glyco means sugar. So glycolysis means I'm going to be breaking down the sugar. Okay, so that's where I'm going to start. So I'm going to break down some sugar. I'm going to make some products in this process that are going to go on to my second main step. My second main step is the Krebs cycle. Okay, that's going to be my second main step. And then I'm going to make some products in the Krebs cycle that are going to go on to my last main step, which is the electron transport chain. These three will happen together. Okay, they'll happen together. Now, they are not necessarily dependent on one another, though. For example, I could do glycolysis without doing Krebs in the ETC. Not for long, as a human, but sometimes bacteria can do that long term. And we'll talk about each of these. But they need the products from the other one to keep functioning. So they can work on their own as long as I have product available. But if my product gets low, I need those other stages in front of me to kick it up a notch so I can keep going. So the electron transport chain can work on its own until it runs out of product, and then it needs Krebs and glycolysis to kick it up. Okay, so we're going to learn how these work together. So in glycolysis, or glycolytic pathways, same thing, we're going to see glucose that gets oxidized. So what's happening to my glucose? Losing some electrons. Now the glucose, depending on which chain we're going down, we're going to learn there's different processes. If we follow it through the metabolism process, it's going to become something called pyruvic acid. Now, basically pyruvic acid is going to be this component that can then move on after a little conversion to the Krebs cycle. Now, the beauty of glycolysis is I'll get a little bit of ATP, and I'll get some electrons picked up by my Uber drivers. So NAD will pick up some electrons that can go on to the electron transport chain. So we're gonna learn how glucose can be used in our cells. Now, there are 3 different pathways that a glucose molecule can go down. The first pathway is the one that we learned in physiology. This is the one that we are most familiar with. It's called the EMP pathway, which is the Embed-Mayer-Perennis pathway. And EMP is our classic. It is done by eukaryotes and prokaryotes when they want to make energy. Okay, this is what most prokaryotes do, what all eukaryotes do. However, we also have another pathway sugars can go down. The pentose phosphate pathway. Did you guys learn about this one in physiology? No, okay. The PPP pathway is going to be one where we can take sugars and we can convert them to create the base components for DNA and RNA, okay? So, for example, if our cell's getting ready to go through mitosis, some of our sugars will be going down the PPP pathway to be used to make new DNA and new RNA for when that cell divides. Okay, so we'll go through the processes of these. Again, since this is nucleic acid production, it's going to be found in prokaryotes and in eukaryotes. Okay, and we'll learn about the process. The last one is the Ettner-Dwardoff pathway, or the ED pathway. This is a pathway that's unique to only prokaryotes. And it's also unique just to a few prokaryotes. Probably maybe about 5% of prokaryotes do this pathway. It's a throwback pathway. Does anybody in here have that one friend that still uses a flip phone? Yeah, we all have that one friend that will not upgrade their technology. They're like, no, my old flip phone's fine, thank you very much, I like it. You know, you're like, they want to get a letter, I don't know, Z, they have to hit that 9 button 3 times to get it to Z. You're like, this is painful, what are you doing? Yeah, you're sitting there texting super fast and they're like, beep, 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 got it. Oh, went too far, hold on, beep, beep, yeah. That's what the Ettner-Dwardoff pathway is. It is the flip phone pathway. It is old school, but the bacteria that do it still love it, all right? They're not moving on. They don't trust this whole EMP pathway. That's too new school. We're not going there. We're fine with our old ways, all right? So it's going to be an old pathway. Yeah.
So 1 and 3 produce ATP and 2 produce.
Yes. So the question was 1 and 3 pathways. 1 and 3 make ATP. Yes. The second pathway, no ATP. Instead, I'm making pentose sugars that are going to get used for nucleic acids. Everybody good on that? So 1 and 3, energy. 2, no energy, but pentose sugars for nucleic acids. Okay, so let's look at our traditional glycolysis. I don't know about y'all, but when I first saw that, when I was a student, I went, ah! And like had a little mini heart attack, right? It's like, well, it's a little scary, it's a little intimidating. So this pathway, we'll go through it in step-by-step fashion. Basically, it's taking a glucose molecule, C6H12O6. It's opening up that glucose molecule and breaking it in half. And what's going to happen is when I do this process, I'm going to generate a little bit of energy. How much? Well, I'm going to generate 2 ATP. And we'll go through this process step by step so you'll get to see where the ATP is generated. I'm also going to generate 2 NADHs. Now I told you when you see NADH and you see FADH2, what do you picture them as being? Uber drivers. So they're picking up electrons. They're Uber drivers. That's their job. And then 2 pyruvates. Well, that's going to be what I break my glucose down to. So glucose is going to get broken apart. It's going to go from a 6-carbon chain to a 3-carbon chain. When it becomes a 3-carbon chain, it changes its name to pyruvic acid or pyruvate. Those are used interchangeably. Okay, those are used interchangeably. Don't worry about the scary picture on the side, all right? You are not going to have to memorize all of those intermediate products. We're going to talk about what I want you to know coming up on a few slides. I'm just showing it to you because we're easing this on you, okay? It's like, kind of like, okay, here's a scary picture and don't look at it. Okay, here it is again. Okay, now we're getting desensitized and then by it's a process. Okay, so the EMP investment. This is where we first start processing the glucose that comes in. And it turns out we have to pay a little price to process this glucose. We have to put a little energy in, a little skin in the game to get the process started. As a matter of fact, when we start the process, we're gonna give up or use 2 ATP. Now that seems contradictory. We want to make ATP, not use ATP. But we have to use a little to get the ball rolling, okay? Then we're going to lyse or break apart our glucose. So we're gonna separate it into 2 different molecules. And then we're gonna get a little energy out. And this is where we're gonna do some rearranging. We're gonna get a little energy out, just a little bit, and then we're going to create some Uber drivers. So let's look at this process. All right, so in this picture, this is kind of a simplified version of what I showed you a few minutes ago. A little less scary, hopefully. We're easing into it. In this picture, all of the little black dots are carbons. Okay, so at the top, we see 6 chains, 6 black dots. That's showing 6 carbons. Now, let's figure out where this is coming from. All right, so what was glucose? C6H12O6. That is a glucose molecule. C6H12O6. Right here, I'm showing you 6 carbons. That came from my glucose, C6H12O6. So here's the deal with glycolysis. When our cells, whether we're prokaryotes or eukaryotes, get glucose, glucose comes in as a little ring. So I have 6 carbons all holding hands. You can picture 6 carbons holding hands, kind of like ring around the rosies. They're all holding hands in this little ring. So what I'm going to do, first of all, is I'm going to have enzymes take this little ring and they're going to open them up. Okay? So I'm going to create a little line of carbons, and that's what we see at the top. That's where we're starting now. We've got a line of carbons. Okay, I told you I have to put a little energy in. Now the reason for this, I don't want this glucose that I just opened to leave the cell. Why would it leave? Well, diffusion, right? It could possibly leave. It can go to a different location. So if I don't want something to leave a cell, what has a hard time getting across a plasma membrane? Large molecules or polar molecules. So if I put something charged on it, is that going to keep it in place? Yeah. So the red balls in this picture are phosphates. So I've opened up my glucose, but I don't want him to leave, so I'm going to put a phosphate on the tail end so he can't sneak across the plasma membrane. That's that first red ball we see. And then, to really make sure he can't leave, I'm going to put a phosphate on the other end, okay? So now I have phosphates on both ends. Now I know for sure the molecule cannot leave, all right? Now, where am I getting these phosphates from? I'm getting them from ATP. So remember I told you I have to put a little energy in, right? So I spend a little energy. So how many ATP did I just spend? 2. 1 for each end, okay? Now, the next step in my process is I'm going to break my sugar molecule apart. Boom, all right? So now I have these 2 ends, where my fingers are, that have phosphates on them, but now I have these thumbs that don't have phosphates. Do I need to add phosphates to those? Yes, notice in the picture, the next step I'm going to do is I'm going to add red phosphates to each end. Where do these phosphates come from? Well, these phosphates are actually ones that are just kind of circulating in the cell. So these are free phosphates. They're not being sacrificed from ATP. They're just free phosphates from metabolism, okay? So they're going to come in and attach to the ends. So I still have only used 2 ATP. These phosphates are freedoms. Okay, so now I have my glucose that's been split into 2 separate 3-carbon chains. Now what I'm going to do is I'm going to have some enzymes come in. And these enzymes are going to undo what I just did. They say, okay, well now that I'm in 2 3-carbon chains, I'm no longer a glucose molecule. My name has changed. I'm no longer a 6-carbon chain. So I don't have to worry about them going out of the cell. So what I'm gonna do is I'm gonna undo what I just did. I'm gonna pull those phosphates off each end. And where are those phosphates gonna go? Well, they're gonna go back onto ADP. Okay, is everybody with me? So I've got phosphates here, and a phosphate here, and a phosphate here, and a phosphate there. They're gonna go and they're gonna get snagged by 4 ATPs. Okay, so how many ATPs have I made right there? 4, right? So I made 4 ATPs. But I told you on the previous slide, we only walk away with 2. But I made 4. So what? But I sacrificed 2. So even though I made 4, I have to compensate for the 2 that I lost in the beginning. So my net total is 4 minus the 2 I lost, which means I walk away with 2. I don't know about y'all, but this sounds really confusing. So an easy way to do this is to go through this together. Everybody make a glucose circle. Okay. So first thing we're going to do is walk to our glucose circle. We're going to open it up, right? Okay, what do I attach to this end?
Phosphate.
Phosphate. What do I attach to this end? Phosphate. Okay, where did those come from? ATP. So that was my use. I had to put a little, skin in the game. Okay, what's my next step? I'm going to break apart. Okay, what do I add here? What do I add here? Okay, now I'm going to do some rearranging of molecules. I just kind of like let my carbons kind of rearrange. And then I'm going to take a phosphate off. I'm going to lose 1. I'm going to lose 2. I'm going to lose 3. I'm going to lose 4. What did I just generate there? 4 ATPs. Now I have 2 3-carbon chains. Okay? These 2 3-carbon chains are going to be what I call pyruvate or pyruvic acid. And they're going to go on to the next step. Alright, so let's try that together again. We make our circle. What's our first step? We open, and then what do we do? Phosphate, phosphate. And then what do we do? We separate it. And then what do we do? Phosphate and phosphate. And then what do we do? We rearrange. We rearrange. Who cares? We're rearranging. Okay, and then we end up with how many carbons at the end? 3 and 3. And then what do we call those? Okay, are we good with this? Yeah, are we good with this? Okay. Because that's how we have to do this to learn. There's a lot going on. Okay, now other things that are going on. Remember how I said we were rearranging? We did our fun rearrangements. Okay, when we're doing our rearrangement, when we're rearranging, what's happening there is carbon's kind of changing who it's holding hands with. Sometimes carbon is holding hands with a hydrogen. It might let go of that hydrogen and grab 2 hands with another carbon nearby. So maybe I'm doing this, and I let go of hydrogen, and I grab hands with another carbon. Yeah. So if I let that hydrogen go, that hydrogen is where I'm going to get some electrons from my Uber drivers. Does that make sense? So when I'm rearranging, another thing that's going to happen is sometimes carbon lets go of a nearby hydrogen. That hydrogen's free. It's going to bump into NAD, and NAD says, hey, you want to ride with me? And bring those electrons, and hydrogen will. And that's how we get our NADH. Okay? Okay, so let's try one more time. Circle. What do we do? Open. What do we do? Phosphate. Phosphate. Where did those come from? ATP. What do we do next? Okay. Where did those phosphates come from? 3 phosphates. Good. Then we rearrange. What do we end up with? How many carbons at the end? 3 and 3, right? What did we generate when we, I forgot, oh my gosh, I forgot. We forgot to kick off the phosphates. Oh, I did the wrong dance move. Okay, let's start together again. Okay, we open, phosphate, phosphate, break. Phosphate, phosphate. Then we're going to rearrange. Then we get to our phosphate off, 2, 3, and 4, and then we have our 3 carbons at the end. How many 3 carbons do I have at the end? 2, and what do we call those? Pyruvic acid. Oh, right there, we just learned glycolysis. See, it's not too scary, right? Wait till we get to the growth cycle. I'm telling you, you're going to like the growth cycle. Okay, so in this process, glucose is being oxidized. It's losing electrons, okay? It is going to be broken apart. It goes from a 6-carbon to 2 3-carbons, okay? When we do that, we create pyruvic acid, or pyruvate, those we use interchangeably. I'm going to create 4 ATPs, but I had to use 2 to get started. So my net total at the end of this is going to be 2 ATPs. Now, the beauty of glycolysis, and remember this is glycolysis of the EMP pathway, is that I can do this really quickly. It's gonna give me 2 net ATPs very quickly. I'm also gonna generate some Uber drivers. I do that when hydrogens are released. This process doesn't require any oxygen at all. So if I'm a prokaryote, I can do this process when I don't have oxygen around. But the problem is I did a lot of work and I didn't get a lot of bang for my buck. I mean, that was a pretty complicated dance and I only got 2 ATPs out of it. So hopefully our next stage will get us more energy. We'll stop here and we'll see how it works out. All right?